Today's webinar is going to be on TCGA Premier. This is a new data product from Biodiscovery. And TCGA Premier is the result of our work in analyzing and then recurating the Cancer Genome Atlas data for copy number information. We've done more to the data since then, and we're going to be talking about it today. Briefly, what I'll cover is a overview of the TCGA project and the data, our curating process and what we've done to improve the copy number profiles. We'll look at some individual samples and some biomarker information. And then my colleague, Dr. Andrea O'Hara, is going to do a short demonstration on a bladder cancer subset where we're going to look at a lymph node involvement across that set on both the curated and the level 3 data. So I think it should be a pretty exciting session. So just by way of background, I think people already know this. The Cancer Genome Atlas is a large project, still ongoing. The total cost has exceeded $375 million from the National Cancer Institute, so it's a very ambitious project. And the goal was to collect data on a large number of tumor samples across a broad range of tumor types. They've collected samples from 32 different tumor types to date, and I think that's going to be the final number. It started with a smaller number and has grown over time. The cancers that were selected were typically selected for impact, so either public health impact or if they were particularly interesting from a genomic standpoint, might even be some rare cancers that were looked at to see if there were any unifying drivers of disease. The tumors have been profiled across a variety of modalities, so whole exome sequencing for sequence variation, copy number through a SNP array, expression, reverse phase protein arrays, methylation, microRNA, and of course all these samples have been richly clinically annotated, which really make these sort of granular analysis possible and really increase the value of the data set. Now, the last point here is the germline analysis. So many of these samples they've also done germline analysis on, in fact most of them, and they either have solid tissue or blood-derived samples to go along with the tumors. This became very important for our work, and I'll talk a little bit about that, but just keep that in mind for now. And just by way of history, you may remember the stimulus program. So this project really owes, I'm just hearing that my slideshow isn't on. So let's see. Okay, now I hear it's good. Sorry about that. So this project came out of the stimulus program, the American Recovery Reinvestment Act. The initial funding was $175 million and has grown from there. So are we seeing the next slide? I think so. Okay. So this is, I love this slide. This is a really nice visual kind of indicating the ambition of the project. And you could see on the right-hand side here the various modalities that were profiled, so mutation information, copy number, gene expression, and so forth, across it has genes here, but of course it's actually whole genome in a lot of cases, and all the various samples. And when this slide was made, there were 12 tumors being studied, and this has grown rather significantly. Our focus, and for today, was on copy number, but we have since integrated mutation data from whole exome sequencing, gene expression data, and of course we look at the clinical data together. So we're going to look at these four axes of data as we go through our analysis, but we're going to start with the copy number information. So when this project was initially released, we were really excited about it ourselves. We went out and downloaded some breast cancer data from the TCGA portal, and when you look at it, this is our favorite software package for analyzing copy number data. It's Nexus Copy Number. And what we found here is some things in the data that stuck out as pretty obvious. So superficially, the large changes in the data set come through. So like, for example, this 1Q gain, very easy to spot. But there were a couple problems. The first is that these small changes were really a mix of some focal changes and germline variants, and they weren't necessarily easy to untangle purely looking at the somatic data. So that was one issue. And you can see it here in a sample. So keep the sample in mind for later. So all these changes here, are they germline? Are there some focal changes? Is there some combination? It was kind of hard to see. And then the other issue is the ploidy. So are the baseline ploidy set right for these samples? And in some cases they were, in many cases they're not. And that in aggregate, when you're looking at 1,000 samples, you can pick up the large changes in the low-hanging fruit, the really recurrent aberrations or high-frequency aberrations. And in fact, the TCGA project themselves, they use an algorithm called GISTIC, which was developed at the Broad. It's really good at that stuff. But once you start getting into smaller and smaller sample sizes, the statistical power starts to evaporate, and these algorithms aren't necessarily as good at picking up these kind of events. So that's where we kind of tried to focus. So again, just remember this top sample here for later, because we're going to come back to it. So looking again, this is still the level 3 data from the TCGA portal, zeroed in here on a biomarker, well-known ERB2 amplification, and here we got a result that was a little surprising, in that 35% of the samples had a gain in this gene. And that is inconsistent with the literature. It's on the high side, where we would expect something in the 20% to 25% range for a sample size this large. And 22% of the samples had a loss in this gene, which again was higher than we would have expected from the literature. So that's just by way of background. Then zeroing in on a particular sample, again, this is looking at data from the portal. Some other things kind of stuck out at us. So one was just the number of changes that were happening, the amount of segmentation on a particular sample, and also the inclusion of germline changes as well. Now, the germline can be dealt with in a number of ways. We took an approach to it. It could also be filtered. There's ways to deal with that as well. But that wasn't the only issue. There are actually a few different issues on a per sample basis that affected the data set. So I'm just going to jump ahead and look at some reanalysis. So this is after we've gone through and done our curating and fixing of the individual samples, and then let's look at that same population profile and see what it looks like. And then we're going to put the two worlds together. So here it is. Again, this is the breast cancer data set. This is now not from the portal. This is what we call our TCGA Premier, or the curated version of the data set. The 1Q gain is still there, although not in as many number of the samples as we saw before. Interestingly, a lot of those small changes have gone away, and a lot of those are germline changes. Now, we didn't just simply smooth out the data, because you can see this focal change in chromosome 7 is still there, and if you go down, look at chromosome 14, chromosome 15p, and also the loss on the chromosome 16 and near the centromere. So we're obviously still picking up focal changes where they're somatic, but we've pulled out a lot of the germline changes, and we've also pulled out a lot of the over-segmentation, which would be false positives. So that's, again, a population-wide profile. Let's zero in on our ERB2 marker, and we see here now we're getting 20% of the samples with that gene amplified, which would correlate to the literature in terms of breast cancer, HER2-positive breast cancer prevalence. So that was all positive. We're happy with that kind of a result. So drilling down to an individual sample, this is now that same sample we saw earlier, but this one has now been, and by the way, the sample was selected at random, and it's very representative. You can look at any of the samples from the portal and then from our curated set, and they kind of look just like this side by side. And you can see here the copy number changes being much more consistent with what we'd expect from working with our cancer cytogenetics customers. So we're seeing whole arm gains and losses combined with some focal events, and we're just seeing a much more consistent pattern. You can almost imagine what the karyotype of this looks like, which would be kind of hard to do with the pre-processed data. The other thing we did, and this is going to become relevant in a few slides as well, is we've integrated the whole exome sequencing data together. So we're bringing two of the modalities together in a single analysis package, which provides another dimension of insight into the data that wouldn't be possible otherwise. So we're not doing anything special with the whole exome sequencing data, except for we're integrating it with the copy number results. So on a per sample basis, you can actually see both the copy number and the whole exome data together. So let's put these two samples side by side. As I mentioned earlier, they look different. I think the sample on the right looks better, but kind of like plastic surgery, you still want to know what's the real natural genomic change that's happening. And so we're going to go and look a step deeper in terms of looking at the raw data that underlie these. So I picked out a few markers to examine. The first is this gain that starts in chromosome 1p and then sort of continues onto the beginning of the Q arm. We'll examine that because it's here in the level 3 data. It's missing from the curated set that we've done. Here's another amplification on chromosome 3q, again present in the level 3 data and not present in the recurated and the curated data set. So what I've done now is I've taken a curated ideogram and I've just laid it on top of the whole genome plot of the copy number probe information for that particular sample. So anyone can get at the level 3 data. You can go into the TCGA portal and download the data. If you go through some approval processes and you have a specific project in mind, you can get approved to get at what they call the level 1 data, which is actually the raw data. And the reason that they have that level of approval is because this data is potentially patient identifying information. So we got approval for our particular project, which was to improve the copy number profiles of the TCGA data set, and they gave us access to the level 1 data to do that. So this, what you're looking at behind it, is the actual copy number probe information for that particular sample. So let's look at those markers that we just talked about. So here on chromosome 1, if you look at the P arm, nothing's happening. There's still that amplification that carries on to the Q arm. So that part of it was real, and you can clearly see it in the whole genome plot. But as far as the P arm, that's a completely deployed region of the genome. There's a deletion here on the Q arm, and that's real as well. The other marker we said was chromosome 3, and again on the P arm, the deletion is there, but the gain on Q is gone, and that again is consistent with what you see in the whole genome plot. So just in case you forgot, this is the level 3 data, and you can see again that one P amplification really doesn't match up. Likewise, the 3Q amplification doesn't match up. Now, you might think this is just a question of ploidy assignment, but it's actually not, because if you look here, this deletion is here and it's real. So it's not just a matter of adjusting the baseline ploidy up and down. It turns out it's both. There's a segmentation issue where the data is over-segmented, and there's a ploidy assignment problem that has to be addressed. So let me just talk about the ploidy assignment briefly in terms of how we address it. So if you've done this kind of analysis before, you know that just with copy number information, you can't really assign ploidy. And in fact, the TCGA project themselves, they switched over from a pure CGH array, which had no SNP information, to a platform that provided them SNP data, which helps in a variety of ways. And one is that we now can do ploidy assignments with the data. So if I add that SNP information in, so this is, again, information that we can't republish, but we can use in our own analysis. So when our curators go through, our scientists actually review these on a case-by-case basis, you know, they will take a sample like this and identify several regions of the genome that are diploid, and they'll line up the B allele frequency of 50-50 A and B alleles with a 0 baseline in the genome plot as a way to assign a baseline ploidy for that particular sample, and then copy number gains and losses are relative to that baseline. So this is that exact sample and realigned to the genome, so you can see that diploidy was indeed an issue, but not the only issue. So what percentage of samples need recentering? If it's just 1 or 2, it really won't matter. It turns out it's a relatively large number in the data set. So for the breast cancer set, it's 134 samples that we actually had to go through and reassign a baseline ploidy for. It does vary by tumor type. Some are as few as 10%. Some samples, some data sets are as many as 50% of the samples had to have ploidy reassignments in order to get that information correct. So let me go back to that level 3 data. I think if you remember the conversation earlier on germline variants and this particular sample. And this is, I wanted to show, this is an example of what happens. So, you know, because we've worked a lot with this data, I looked at this and thought this is probably just, these are probably germline events. So a breast cancer sample, maybe it's mutationally driven. And in fact, when you do the cleanup and the recurating and you look at the events, most of the copy number events go away. You're left with 6. They're all focal. And you're left with a large number of mutation events. Now, I haven't gone deeper on this sample, but it looks mutationally driven. And if I were doing copy number research, I might pull this sample set out if this is a class of tumor that's being driven by mutation, not by copy number, which would improve the statistical power of my copy number set. And conversely, I might include this in a sample where I want to look at only tumors that are driven mutationally. So having the data together really allows you to do this plus other things that you wouldn't be able to do with 1 or 2 of the modalities by themselves. So here again is the before and after picture. And so how do we get from before to after? So I showed you the population profile. We went through in a particular example, and I think you can see the improvements in the copy number profiles that were achieved. How do we do it? So what's our process? And I think this is important for people to understand. If you're using this data for research, well, what is it that we did with the upstream data to make it more valuable? And a few things. So this is a very simplified view of our process. There's actually much more to it than this, but these are the highlights. So the first is our mechanism for addressing the germline polymorphisms is to use the blood-derived pair match samples wherever possible. So we'll use the solid tissue samples if the blood samples aren't available, but the solid tissue samples are more likely to contain tumor DNA as contamination. So we tend to use the blood-derived samples where possible, and that removes the sample-by-sample polymorphism. So it's not just like a simple filtering against DGV or something like this. We're actually doing it on a per-sample basis. The second step is once that's done, we analyze the entire data set and we resegment it with the Nexus copy number SNP-Fast2 algorithm, which is an HMM that's really been optimized around cancer samples. So this is a hidden Markov model. I'm not going to go into depth on the model today, but it uses both SNP probes and copy number information in assigning states, and it has a variety of intermediate states, not just the 5 standard states, for assigning mosaic events, and it's really effective at untangling the complexity of cancer samples. Once that's done, so far we've just done algorithmic work, and we've learned that that's just not enough to get to the level of data integrity that we'd like. And the gold standard for this kind of analysis is actually for a trained scientist to review each of the samples. So that's what we've done. Every sample is inspected on a whole genome basis. We manually call low-level mosaic events. So we could adjust the algorithm to kind of pick those up, but then you get a bunch of false positives. So we leave the algorithm set in such a way to maybe flag those, and then we'll manually call them. Baseline ploidy assignment I've already discussed. Where that's ambiguous or a sample has a really complex genotype, we'll just flag it and we'll annotate it. Likewise, we'll find chromothripsis events and we'll annotate those as well. So that's something you wouldn't normally get. After this, we take samples that have poor quality or an ambiguous matched germline, and we just pull them out of the data set because they really just contaminate the data. After that's done, a second trained scientist reviews all of this, and it gets published up to NexusDB. So unless you have more questions on the process, that's all I really wanted to say about it, and we'll go back and look at some more data. So here's an example. I mentioned that we'll highlight complex genotypes like this, and if you can figure out where the baseline ploidy is, please send me an email because there's really no way to do it as far as I know. However, it's still a useful sample. The copy number events are real. The events are relative to one another, so from a dosage standpoint, this might be important. The ploidy is ambiguous, but that's okay. We flag it, so it's annotated in your data set as complex genotype, and you know that this ploidy may be ambiguous and may be incorrectly set. Complexity itself can be prognostic. In many cancer types, just having a complex copy number profile itself can be an important indicator, so that's another reason why having it annotated is really important. Fortunately, not too many samples fall into this category, but there are a few, and these are poor quality samples. They'll often have no copy number events being reported, although you don't really know what's going on because you really have some upstream problem with the sample. It could be hybridization or who knows what. But in these cases, since they're not adding any statistical power to the data, they just get removed because all they're going to do is contaminate your studies. So there aren't too many of these, and we can provide lists of the ones that have been pulled out, or you can just compare it against what's in the TCGA. But we've removed those that have poor sample quality. So just want to shift gears, look at one more tumor type, and then we're going to do the bladder cancer analysis. And I want to show you this because I also want to show you the chromothripsis and the loss of heterozygosity and how we're dealing with that. So this is a kidney chromophobe cancer type. It's a kind of interesting cancer because it's hypodiploid. It tends to have these deletions that have been fairly consistently reported. It's rare, it's relatively indolent, but nonetheless it's interesting from a genomic standpoint. In the level 3 portal data, we see the deletions clearly, but we also see these high-frequency gains. And so the question is, again, which one is more consistent with what we'd expect from kidney chromophobe? And indeed, it turns out that it's the curated data, unsurprisingly, is providing results that are more consistent with the literature. And so you can map, again, the hypodiploidy to this paper that was published. This is, again, a rare cancer. There aren't that many samples out there, but the profile is pretty clear. These stars in this paper indicate copy number loss events, and you can see it very closely matches the frequency plot that we've seen by analyzing the data. I mentioned chromothripsis earlier, and there's a nice example within this small data set of a chromothriptic event. So this particular sample, you see most of these samples just have these whole arm deletions or whole chromosome deletions, so hemizygosity, pretty consistent. And then all of a sudden you get to chromosome 8 for this sample, and it's just a mess. And this is a chromothripsis event. So we recognize it and we annotate it and flag it. And this is really, I put these two up side by side so you can see in the left-hand side, once we've recurated and analyzed the sample, again, you can almost visualize the karyotype in your head, and you see chromosome 8 as a nice example of chromosome shattering right here. Looking at the level 3 data, it's not really clear because chromosome 3 might indicate the same thing and so forth. And this, again, is really mostly because the data has just been over-segmented on a per-sample basis. So it would be very hard for someone to quickly identify these kinds of events before having done this analysis. Last example, I promise, and we'll move on to data. This is a colon cancer, and this again is data from the portal, and just wanted to quickly go through it. So chromosome 2, again, you have the over-segmentation, lots of gains and losses, but they're all focal. Just keep this in mind. Chromosome 5, you pull it out here, it's over-segmented, it's probably trisomic. And then you have this very high gain in chromosome 13, 4 copies, so tetrasomic event on chromosome 13. So that's what's in the level 3 data. Now, this particular sample, once we've recentered it for ploidy and resegmented it, you get a very different look at it. So remember chromosomes 2, 5, and 13, and now the picture is very different. So this particular sample's chromosome 2 is not actually a normal with a lot of small changes. It's actually a relatively quiet hemizygous chromosome. Chromosome 5, which was reported as trisomy, is actually normal, and 13, which was tetrasomic, is now trisomic. So you can see this really radically changes the interpretation of this particular sample. And when this affects a large number of samples in a study, unless you have very high frequency recurrent events, you're not going to find these sorts of changes. So I think we've made this point. This is sort of the before and after again of this colon cancer sample. And I mentioned earlier how we're using the SNP probe information to assign baseline ploidy. Another thing we can use the SNP probes for is finding regions of homozygosity. So although we can't publish the SNP probe data because it's identifying information, we can manually call the regions, which we do, as LOH, and then publish that. So if you're analyzing the sample and you look at the copy number track, there's nothing happening. But on the zygosity track, you see there's this nice region of LOH, which again could be indicative of something. So we flag that out. And in this particular case, because we've integrated the mutation data, you'll see that the LOH region actually is also overlapping with mutational events in the allele. So this is a case where you might have had a mutation on one allele and it got replicated, and you wind up having LOH on both alleles. And if it's a tumor suppressor and it's inactivating, that could be a driver of disease. And this is something that would be very hard to pick up otherwise. So with that, as an introduction to what the data process was for us, and hopefully gave you enough of a flavor of the per sample quality in the TCGA curated data set that we've released, I'd like to turn this over now to my colleague, Dr. Andrea O'Hara. She's got a very nice example exploring a bladder cancer set. We're going to look at some data live together and profile it. So with that, I'll turn it over to Andrea.
Great. Thanks, Louis. Thanks for that introduction. So to get started, I first want to show you how we can access and download the TCGA data through NexusDB, which is our online repository. When you first launch this, you'll have the option to log into NexusDB right from the front. If you already have Nexus open, you'll have this NexusDB tab that's available. First-time users, you can register a new account. If you already have an account, you can just log in, and you're going to click on Visible Projects. Now, when we click on the visible projects, we're going to see all of the data sets that are housed within NexusDB. If I sort by the name by clicking on the header, we can actually now see all of the different data sets. For this particular analysis, we're focusing on our bladder cancer or BLCA analysis. I've already taken the time to download it for us today. So let's flip over to that by clicking on our data set tab. This project consists of 205 samples. It's well annotated by the TCGA data, so we actually have a number of factors that go with it. This includes the comments that we've added in, so the curator comments, as Louis mentioned. We also have the clinical annotation from the TCGA, so this includes things like pathologic states for nodal involvement, metastasis, risk factors, tissue site, etc. So with this bladder cancer project today, I want to take you through three different parts. The first thing I want to do is show you how we can actually use this data for both discovery and or validation within our own interests. And then I want to show you guys just very briefly how this curated data differs from the level 3 data downloaded from the TCGA data portal, and finally how it compares to the marker paper published earlier this year. So to get started, I'm just going to go ahead and click View. Again, we're looking at the 205 samples that have been curated and were available on our TCGA Premier project. Now, when we look at the data here, okay, this is showing us a higher level overview. The top is our aggregate or summary overview. We can see blue to indicate gain, red indicates losses. Again, this is showing us for all 205 samples right now. We also have each of the individual samples, it's shown in blue down here for the name. Again, blue for gain, red for loss. We also have the alleles calling, and as mentioned earlier, we have these areas that are loss of heterozygosity, which may or may not correspond to copy number alterations. And again, this is data that we're not going to see at all in that level 3 data from the data portal. Now, when we look at this from a higher level, and I'll go into this later, we find that on the whole, this is going to be somewhat similar to what we would find reported in the marker papers. But again, those tended to focus on the higher level view. When we start to drill down into different subtypes or subgroups within the sample set, that's where we're really going to see the value in our TCGA Premier data. Now, for me in particular, I was interested in focusing on lymph node involvement. So this is one of the annotations that was included within this particular TCGA data set. And in particular, I was very curious to do a little bit of discovery looking at samples that have no regional lymph node involvement. So the first thing that we can do is we can actually mark out which of these samples have different pathologic end scores. So this is the nodal involvement score. The groups, just briefly, while I get this going, the breakdown is N0, which is no regional lymph node spread, N1, one lymph node involved, N2 is multiple lymph nodes involved still within the pelvic region, N3 is multiple lymph nodes to the common iliac artery, NX is not enough information, and then we have some that are just blank, which are unclassified. Both of these two groups are pretty much non-informative for us, so we can actually zoom in further and exclude those from our view. Now, you can see here when we're looking at these aggregate groups, we have a different number of samples for each group, but again, we are looking at some sort of an aggregate. It looks like they're overall fairly similar to one another. We can see there's sort of common trends. We see this like 5P, for example. Gain is frequent in many, whereas the 5Q loss is also frequent. But it's hard to tell when we're just looking at this with our naked eye if there is anything that's going to be discerning, say, our N0 no nodal involvement group from our N2 multiple nodes involved group. And I myself wanted to know if we can actually distinguish this N0 group from the other groups with statistical significance. Now we can actually test this with Nexus using our comparison feature. This is going to run a Fisher's exact test between two groups. Here I selected the N2 and N0 groups. So I can see if there is a statistically significant difference in terms of the copy number profile for our no lymph node spread N0 group and the multi-lymph node group. I'm going to switch right to the genome view, which very clearly shows us if there is a difference or not. So we can see here we have, again, 24 samples in our N2, at least 2 lymph nodes involved versus 87 in our N0. We can see the profiles just as we saw the previous screen. However, we can see areas that are considered statistically significantly different between these two groups. And what we can find is that the ones with nodal involvement have significantly higher frequency gains of 1p, 2p, all of chromosome 3, chromosome 7, this 13q terminus, mid area of 17q, or sorry, 15q, 17q, and a little bit of 19q. There's also additionally some focal changes that are discriminating these two groups. So I could choose to look at these more carefully, but again, at this point, I was really just trying to validate that this N0 no nodal involvement group is a distinct group from the other different node subgroups. So with that in mind, I wanted to learn more about just this N0 group. Now what I can do is I can actually go back to my data set tab here, and I can sort based on our nodal involvement and select just these subset of samples. So I'm just sorting based on that annotation factor, and I'm going to select just the N0 group. Now again, if I click View, I'm going to get that aggregate overview plot, but this time we're going to be focusing in just on these 87 N0 samples. Now, if I wanted to find out what some statistically significant peak alteration regions of change are here, I could run a logistic analysis. But I think that within this subset, I'm not necessarily sure that's the route I want to go. I'm really more interested in survival. So we can see here that there's a lot of different copy number changes, and we would anticipate that this N0 group with no lymph node involvement would have, on the whole, overall better prognosis. My question with this particular group was, are there any copy number changes or allele changes within this sample set that indicates poorer prognostic outcome? So are there individuals that should have a better outcome? Do they have any changes that would actually indicate poorer prognosis? There's a tool within Nexus Copy Number where we can actually ask that exact question. And this is called Survival Predictive Power. All of the TCGA data does have survival metrics associated with it. And so if I go to our overall survival column, again, I've just selected only this pathologic N0 group, and run Survival Predictive Power here, I can actually ask the software to identify statistically significant regions of alteration that have an impact on survival within just this subgroup. So I'm able to quickly see if there are any areas that do affect survival within this limited sample set. Now, as you can see here, that was a really fast response, and we can see that there are several regions that come up. I'm going to change this to a combined view so I can really see an overall larger picture. And I'm going to sort by the types of changes that I'm seeing. You can see here I have several areas of allelic imbalance, areas of copy number gain and copy number loss, and I also have several areas of LOH. Now, as you'll remember when we were talking earlier, this is going to be unique to this TCGA Premier data set because we don't actually have the LOH column. We'll look at that level 3 data directly from the data portal. Now, when I was looking at this, I saw that there were 3 different groups of LOH. I actually don't know very much about any of these regions. You can see one of these is, in fact, it has 7 genes here on chromosome 9 that seems to have a fair number of samples here in this in-group that have this region of LOH. So my first question was to just take a look at these samples. I was curious to see, are these copy-neutral LOH or are these associated with gain or loss? So what I can do is go to my Results tab and just view these changes. I'm going to change our view to be the allelic ratio and sort. And so we can see that we have several samples that have a copy-neutral LOH at this area. So this is an area, a stretch of homozygosity that's not associated with a copy number alteration. This happens in cancer samples and usually means that we're targeting some sort of gene of interest within here. So that looks like that could be pretty interesting. Now, to follow up with that, I can actually take a look at this and say, well, how does this actually affect survival? and look at the Kaplan-Meier plot, I can simply highlight it here and click Add as Factor. And if I go back to our main data set tab here, I have, it just popped right up for me, LOH of this region. If I right-click and click Kaplan-Meier plot, I can now here very quickly see what this survival plot looks like. So if I hover over this, we see that if we have loss of heterozygosity at this region of chromosome 9, we have very poor outcome versus those that do not. And again, we're looking only at these ones that have no lymph node involvement. So they should have overall a very good outcome. And we can even assess the statistical significance. So we've done a lot of discovery now with this data set. We can actually also use our TCGA data for validation. We can do it within a data set that we have here, or if we were looking, let's say, at our own data set that we started with. So what I can actually do is if I go back to our predictive power results, I have this option in here to query database. Now when I click on this option, what I'm going to do is I'm looking at all of the other TCGA Premier projects to see if this region of chromosome 9 has been marked in any other samples for loss of heterozygosity. This is a way for me to validate if this change is something that's occurring in other data sets and with what frequency. From there, I can start to delve even deeper into these data sets to see if it also is associated with lymph node involvement, if it's associated with an outcome in survival analysis. I just want to take you through what those results look like here. So it starts off first returning the projects that have this change. There are numerous projects within the TCGA Premier set. 16 of those projects had samples with LOH within the region. We can see the total number of samples, the samples that actually had that change, and what that frequency works out to. So we can see that perhaps it's more frequent in some, but less frequent in others. If we want to look at which of these individual samples that pertains to, we can actually click on the samples page. Now when I'm in this option here, I can now look at all of these different samples and any of the annotation associated with them to see if there are certain ones I'd like to select. Perhaps I want to download those samples that have LOH to include in a meta-analysis. I can do that right from the screen here where I can select multiple samples and then download them. I can also take a look at what these calls are. This gray area here is our query region in question. And we're looking at this larger window is the entire P arm of chromosome 9. If I sort, I can see that there are numerous samples that have basically LOH of the entire chromosome arm, but we also have several samples that have a more focused region of LOH, and these ones might be the ones I'd be more interested in evaluating further. Now, as you can see here, this is a way that we can use the TCGA Premier data set to validate our original findings. So stepping back a little bit, I'd like to just briefly take you through how this data set compares to some of the other data sets, so you have the peace of mind of knowing that we're starting from a good place. So we looked at, when we looked at the overall data set here of all of the curated data, we saw that we had a nice higher level overview of what was going on within the data. I'm just going to reselect that and give it a look. We can ask ourselves how this compares to the marker paper that was published earlier this year. Again, the marker paper typically focuses only on these higher level changes. And so when we look at the data set as a whole group, what we're going to see is that on the whole they're very similar. So if we look at this aggregate plot at the top here, we can see that in the marker paper, they reported a high frequency of amplifications on chromosomes 3, 5p, 7, 8q, 17q, down here, 19q, 20q, and 21. And this black bar here is indicating our threshold of 35%. And we can see that we do reiterate that they are frequent, though the percentages may vary slightly. When we look at the frequent deletions that were reported, again, at just 35% frequency, it included for just the P arm, all of chromosome 5, 6q, 8p, all of chromosome 9, 10q, 11p, 17p, down here, and 22q. And you can see, again, we have some variation in frequency. Again, not surprising because with diploidy correction, that is going to have an impact on the frequency, but we can see that the trends are holding up overall. If we wanted to run logistic analysis on this to identify those statistically significant frequent changes, we could do so within either this larger set or, again, the smaller subset. If we do logistic on this large set, we can go to our main data set tab, click on Tools, click on Logistic. When we actually look at the results, right here, we're going to get a table with our regions, our extended regions of change, and then our statistical significance. Now, logistic functions to identify the highest peaks of the mountains and the lowest valleys in terms of our aggregate copy number changes. So it's those that are very frequent, very focused, with added weight for the high amplitude changes, the gains, multi-copy gains, and homozygous deletions. When we look across our results from the marker paper and from this curated data set, we find that on the whole, these are nearly identical. The curated data set comes up with a few more results, but really they are very similar. And again, not surprising because we're really focusing on those super high level changes, which are the same for both. Now, what if we actually compare this to what we would get if we were looking at just the TCGA data portal results? I have included that in this data set as well. And you can see here, I can select multiple data sets to view all at once. You can imagine when you're doing your own analysis, this is the way that you can choose to integrate the TCGA data with your own analysis if you so choose. Here I'm integrating the curated data with the level 3 data portal data. So when I select for all of the samples here, I'm going to add a factor that's just our data source, so we can compare those that were downloaded from the curated data set versus those from the TCGA data portal. Now what you can see when we look at this aggregate on the top, I don't know if you recall what it looked like before, it looks a little more whiskery perhaps than it did earlier. If we take a gander at our individual samples, the top is showing us those reprocessed curated samples. The bottom here, I just scroll down, is showing us these level 3 segmented samples from the data portal. And you can see we can break this up into roughly 3 different groups. We have some samples that have a lot of very small focus changes, but not very large changes. We have numerous samples here that have what looks like almost the entire genome with copy number alteration. And then we have some samples that are kind of in the middle, so some of these focus changes, as well as some whole chromosome arm changes. We can actually compare these as a set, just like we did earlier with our lymph node involvement groups. So we can compare the curated versus the TCGA level 3. And what you can see here when we do this just brief comparison visually, we can see that, again, the trends are holding up. That we can see, for example, this large gain on 5P, loss on 5Q is maintained. However, we do have a lot of this whiskery focal noise, more or less, which might lead out some of the more interesting focused true changes that we can see here, like, for example, this loss on chromosome 9. So I just have taken you through sort of different parts of this data set. So just to reiterate here, we can use the curated TCGA Premier data sets for both discovery and validation. We see that it compares fairly well to what we get from the marker paper, which is giving us really just a higher level overview, so that is reiterated within the data set. However, we do see that it is much improved, the curated set versus the level 3, in terms of our overall quality of scoring, which can help us to identify some of these focal areas of change. And with that, I'll see if we have any questions.
Okay, Andrea, thanks very much for that great overview. It was really nice to see the bladder cancer set profiled. So again, if you do have questions, you can enter them into your GoToWebinar panel. There is one question that we had earlier that I just want to re-ask here, which is, how do we decide on the sample ploidy, and do we have any other orthogonal comparisons like FISH for maybe FISH annotations for some of this data that we can compare to?
That's a great question. So Louis alluded to this earlier in the introduction. When we talk about assessing where the baseline should be set, it is a very painstaking process. We're taking into account the entirety of the SNP array data for each individual sample, along with any knowledge that we might have about that particular tumor type. And we're using that to assess what the appropriate baseline will be for that sample. Once we've assessed it, we actually have both a secondary and tertiary review of our baseline regions to ensure that multiple scientists agree on that baseline setting. Now, unfortunately, we don't actually have access to these TCGA samples to run our own FISH analysis, but there are some of these data sets that do have FISH analysis that's been run on them. And we do find that when we look at the curated data afterwards, we have a very high level of concordance with any FISH results that may be reported. So that does speak to the fact that we are going in the right direction.
Great. So here's another question, which is an interesting one, are any pathogenic germline changes found that are then kept in the curated data?
That's a very good question. So the way that our data is currently set up, we're actually focusing only on the somatic changes. Because we're doing a matched paired analysis, any germline changes are being removed from this particular analysis. That being said, there are some clinical annotations that are kept throughout that may indicate if they have any pathogenic germline changes that are already known for a certain disease. So, for example, if they, I believe in one of the colorectal or colon sets, if there are certain risk factors associated with it that are pathogenic, that has been noted. But on the whole, we're really focusing on somatic changes currently within these data sets.
Great, thanks. Okay, I think that's it for questions. I do want to mention, because I know we have some users, and this came up before, if there are users, places at MD Anderson Cancer Center and at the National Cancer Institute already have access to the software and to this data. You just have to contact the right core lab to get access. That's already, so there's no cost for an individual user to get access at least those two centers. There are other centers that do have site licenses for the software and for the data. So if you're a center that you're not sure, just email us and we can kind of check with who to contact within your center.